Hello everyone, my name is Thomas Catilla and I'm a master's student at the Institute of Plant Breeding, Genetics and Genomics at the University of Georgia. Today, I'll be showing you how to conduct gel image analysis for semi-quantitative RTPCR. This process uses band intensity from a gel image to quantify relative gene expression. In order to begin, you'll need first an image of your gel, second, ImageJ, a free and publicly available software for image analysis, and third, a spreadsheet program such as Excel. So here I'm starting with an image of my gel opened in ImageJ. To analyze your gel, just use the rectangular select tool to select the first band in your gel. Once you've selected that, go to Analyze, Gels, and select First Lane. Next, just drag the rectangle over to the second lane in your gel, and then go to Analyze, Gels, and select Next Lane. This can also be done with the Command+2 shortcut in Mac. So we'll continue on doing this until we've selected every lane in our gel. Okay, so now that we've selected all of our lanes, we can go to Analyze, Gel, and Plot Lanes. This gives us a graph of every peak for every lane that we've selected, and now we can just use the straight line tool to go ahead and segment off those peaks. Okay, and now that we've marked off all of our peaks, we're ready to go ahead and measure peak area. So all you have to do is select the wand tool and then select each peak that you've segmented off. And you'll notice as you do this, ImageJ will automatically record all of the peak areas in a pop-up window in the order that you click on them. And so just go ahead and move through and do this for every single peak. And now that you've done this for every peak, you'll have a data set that you can go ahead and copy and paste into Excel. And so here you can see a larger data set that I've already gone through this process for and moved into Excel. And notice that I've also gone ahead and labeled the template or the cDNA that I've used and the primer set that I've used for each lane. In this example, I'm comparing 3 control samples to 3 samples that have been exposed to a salt stress for a control gene and for 4 different genes of interest that I think might be differentially expressed. Now, in order to make a more objective comparison between our control samples and our salt samples, we need to calibrate our genes of interest to our control gene. Now, this allows you to control for some of the overall differences in cDNA content between the libraries that we prepared for each individual sample. So I'll just go ahead and plot this as the measured peak intensity for our gene of interest for sample C1 divided by the peak area for the control gene for the same sample. And we can just go ahead and do this for every one of our samples. So control 2 in our gene of interest is being compared to control 2 in our control gene. Right, and now you can see that I've gone ahead and calibrated all of these, and now we're ready to go ahead and take an average for all of our 3 samples in each gene and for each experimental group. So I'll go ahead and do that now as well. Alright, so what I've gone ahead and done here is taken the average of all of these calibrated values, and I've also moved them into their own table so that they can be more easily plotted. So let's go ahead and select this data, and we'll go to Insert Bar Chart, and let's also go ahead and add our standard error bars. So now we're able to look gene by gene, comparing our relative expression levels in the control group and the salt group. Quickly, I just want to note that for the control gene that we use to calibrate these, they're categorically at a relative intensity ratio of 1 because they're divided by themselves. Now going gene by gene, comparing the 2 groups, we can see that the only gene that's differentially expressed is gene 3, and that appears to be upregulated in the salt group. So that's how you can use a gel image to quantify relative gene expression for semi-quantitative RTPCR. Again, my name is Thomas Catilla. Thank you for watching.